Hi, our project was developing a nanoladder scaffold for neuronal outgrowth and regeneration. Spinal cord injuries can cause drastic changes to the body, including loss of function and diminished function at or below the site of injury. There are currently approximately 253,000 sufferers in the US alone, as well as an additional 11,000 each year. Unfortunately, for those suffering from spinal cord injuries, there are only palliative treatments available. Some of the leading causes of spinal cord injuries are vehicular accidents, falls, sports accidents, and other forms of violence. The reason that these injuries are so difficult to treat is that there are significant barriers to spinal cord injury healing. The first barrier is called the secondary injury. The secondary injury is a cascade of biological phenomena that occur after the primary injury, including scar formation, tissue loss, and cytotoxicity, and all of these things combine to make it really difficult for neurons to regenerate and reform functional connections. The second critical barrier to spinal cord injury healing is that there's no directional growth cues existent at the site of injury. So in order for the neurons to regenerate and reform functional connections as necessary, there are cues present to allow them to grow and reform their functional connections. There's been some previous work developing scaffolds that could both grow and align them properly. These generally involve looking at topographical features on scaffolds on both the nano and micro scale, as well as different chemical features. So that brings us to our goal for our project, which was to develop a nanoladder scaffold, which does incorporate both topographical features and chemical cues. The topographical features include micron-wide stripes that run along the length of the scaffold, as well as nanoparticles that are spread over the surface, and we also incorporated chemical cues using extracellular matrix proteins. So, for our central aims for this project, the first one was to compare neurite elongation on various extracellular matrix proteins. So the goal of this was to study different proteins and see which ones would elongate neurites the best, so that we could find out which would be the best to include in our final project. The second central aim for our project was to develop the silk nanoladder fabrication process. So previously our lab did not use silk to fabricate them, but we switched over to silk because of its biocompatible nature, but that meant that our group had to devise a fabrication process in order to get the stripes and nano protrusions as well as a chemical coating on a silk substrate. And the third central aim was to study how the neurites grow on the silk compared to the previous nanoladder to ensure that the silk actually enhances the neurite's ability to survive and grow and also align on the silk nanoladder, and we wanted to make sure that for whatever reason they didn't actually inhibit the growth of the neurons. So for our approach to one of our central aims, which was the extracellular matrix protein coating, we took our silk scaffold and sterilized it, and then we prepared 3 different protein solutions, laminin, poly-D-lysine, and fibronectin. Next, we coated the scaffold in the extracellular matrix proteins, incubated them overnight, and washed them several times in order to wash away any excess. Our approach for the cell culture and imaging involved obtaining embryonic cortical cells from rats, and then we seeded the cells on the scaffolds and incubated them for 7 to 10 days roughly. Then we fixed the cells, used anti-tau staining and imaged them, and then we used an ImageJ plugin that would enable us to both analyze the directionality and also the lengths of the neurons. So for part of our results, we looked at how the silk nanoladder influenced the directionality of the cultured neurites. So you can see here with the control that there was very little alignment in the regenerated neurites, and our control in this case was just plain silk without any topographical features, no chemical cues, just plain silk. And then for our nanoladder, which did incorporate both chemical cues and also the topographical features, you can see very clearly that there was significant alignment. So this was a really positive result because it showed that we were able to align the neurites using our scaffold. So the next significant piece of data was we studied neurite outgrowth on silk. So we looked at 4 things. Our control is just plain silk. Our nano protrusion part here was just the nanoscale topic.